Now that you have seen the next generation sequencing video, we will do a demonstration of how the DNA is prepared for this sequencing. This is called the library preparation. Once we have extracted the DNA, we need to fragment it. Why? Because, as you have seen in the video, the fragments of DNA that attach to the flow cell needs to be of a specific size. We also need to prepare this fragment to attach to the flow cell. A flow cell looks like the one in the image in the top. In reality, we will make millions of small fragments of DNA to attach to it. The library preparation consists in fragmenting the DNA and adding adapters that will make them bind to the flow cells. There are different ways to do this. There are different chemistries. What we will be demonstrating today comes in a kit called Nextera DNA Flex Library Preparation, and it uses a kit that's a chemistry based on bead fragmentation. The steps that we are going to show in this video are: first, genomic DNA fragmentation. In this step, we will be fragmenting and tagging the DNA at the same time, using magnetic beads, which have transposons linked to it. This is done with a fragmentation reaction, which involves a transposon cleaving and tagging the double-stranded DNA with a universal overhang. After this step, we clean the DNA that was not binded to the beads, and other reagents are washed away in this cleanup step. In the next step, we add the adapters that are going to make the fragments bind to the flow cells. These adapters are also used as primer binding sites to amplify these fragments that we have created. Because we have created DNA fragments of different sizes, we need to select the ones that will attach to the flow cell and adequate for sequencing. The size selection process for this kit is done with magnetic beads. After this, our library is complete and ready for quality control and sequencing.
Hi, my name is Kay Anantanawat. I'm a manager of the UTS sequencing facility, and today I'll be showing you how to do the library for the Illumina sequencing platform. And the library prep is called Nextera Flex.
After the extraction and before library preparation, we need to measure the DNA concentration in our samples. We can do this using Qubit, which is a fluorometric-based method.
First of all, you need the genomic DNA, which you want to make the library form. So with the Nextera Flex kit, you can actually use it to make the library for the whole genome sequencing. So basically, it's a bacterial isolate, or you can also use it for the metagenomic, which is so the sample that, for example, like environmental sample that have millions of bacteria, and just use the bacterial, the extraction. So first of all, I will move this away. The kit itself composed of like 3 boxes, and it has like 3 different components, but the most important part of the Nextera Flex is the bead links transposon. So this bead is the core of the of this type of library prep. So basically, this bead is a magnetic bead that contain transposon and the transposons sequence. And what it will do is that this bead will actually go into the genomic DNA, cut the genomic DNA at a particular point, and then inserting the transposon sequence. So the fragmentation process composed of the BLT beads, genomic DNA, and the buffer. So the fragmentation buffer. Yeah. So the protocol for the Nextera Flex is published online from Illumina website. So if you are more interested in the detailed protocol, then you can have a look at that. So first of all, you need to spin all the tubes to make sure that like any liquid is going into the tube. So what we will do is we will use the a mini spin So spin down quickly. And then after that, so for the segmentation buffer, vortex is thoroughly for use. And then again spin. So for the beads, we don't vortex for mixing, so we use the pipetting. So we use pipette to do the mixing. So currently the bead is separated from the liquid. So we need to fully resuspend the bead. So this is done by pipetting like a few times. So now the bead is fully suspended. So the reason that I don't vortex this is because like if you vortex the bead, some of the bead will get stuck on the lid, and then if you have to spin it down, then the bead will separate from the liquid again. So that's why we use the, because the segmentation will be done in the thermocycler, so that's why we need to use the PCR tube or 0.2 ml tube. And we gather the tube. Nanogram of DNA into the segmentation process. And the volume for the DNA is 30 microliters. And then transfer 10 microliters of TB1. And then 10 microliter of fully suspended beads. Make sure that there's nothing left on the tips. And then use the P200, adjust to 30 microliters, and then use it to mix the beads in the tube. Again, the reason here, instead of vortexing, I don't want to vortex it because I don't want the beads to actually get stuck on the lid and I have to spin the tube. So that's why I use the pipette to suspend. And then we just put the tube in the thermocycler. And the thermocycler is now set to the segmentation process.
Step 2 is segmentation cleanup. We will need to add a reagent to stop the segmentation reaction, and then we're going to remove all DNA that is not binded to the beads. Okay. So the tubes, so they are now cooled to 10 degrees Celsius?
Yes, yep. So we can take it out, and then make sure that like you spin the tube first, so that like any liquid that got evaporated and get onto the lid can actually be spun down to the bottom. Then we need to add the stop reaction, a stop segmentation reagent, and that is, so as you can see, it's now
no precipitate.
Yep, there's no precipitate. Yep, it's just a quick spin. we add the 10 microliters of the stock solution into the reaction. Again, we resuspend the reaction using pipette. So as you can see, it's separated from the solution. So the bubble is normal for this because the stock solution contains like SDS, like the foaming detergent. The thermocycle and incubate at 37 degrees for 50 minutes. In the tube out, and again we spin it first. It is always a good routine to spin before you open anything. And then now we need to wash the beads. So to wash these beads, this is the magnetic stand for 0.2 ml tube. So it can be used for PCR tube like this, or a PCR plate like this. So the way that it works is that just put the plate onto it, and in this case, in our case, we're using the tube, so we just put the tube on it. And what it does is that it actually pulling down, pulling the beads to form a circle on the wall. So that you start seeing that band. Yep. So we're going to leave that for about a couple minutes, just to make sure that like, all the beads are separated from the solution. Okay. So it looks like the magnetic beads are separated from the solution. So we just need to remove the supernatant. So carefully put the pipette tip in the middle of the tube and not touching the wall, because you don't want to disturb the magnetic beads. Slowly pipette the supernatant out, and then make sure that like before you discard the supernatant, that is there's no magnetic beads in the tip. Cool. Yeah. And then now the bead need to be washed by the TWB 3 times. So fully resuspend the beads. And then leave it on the magnetic stand again. So now what is in the reaction at the moment is the magnetic bead or the BLT bead that on the surface of the bead containing the genomic DNA that has been segmented. This process we should only left with the segmented DNA. So we wash the bead with the TWB 3 times. When it comes to the last wash, just leave the beads in there, and then we go and prepare the reaction for library amplification. So the library amplification is the process where we add the barcode onto the library. So for example, if you have a 96 sample, this is where each sample will be tagged with a particular barcode, so that you can pull it together and then sequence. So to do that, we need to create a PCR reaction. So first we need to add, so the recipe of how to make the master make is in the Nextera Flex protocol. So that is the PCR master mix.
Okay.
Yep. So again, we spin before we wash it. We add 20 microliters of the EPM. Now we remove the supernatant, the last TWB wash from the sample. Then we quickly add the master mix that we just made. So don't leave the bead to dry out. So fully we suspend the beads in the mix. And now we add the barcode primers. So we have two barcode, two primers. So I7 and I5. So I7 is a forward and I5 is a reverse one. So this will tag. We'll add the barcode on both ends of the library.
The PCR is to add the barcode and to amplify?
Yep.
Okay.
That's right. So again, use the pipette to mix. Then we put it in thermocycline.
So in this final step, we will clean the library and select the fragment size, which is the most adequate for sequencing in the Illumina flow cell. So we will use SPRI bead size selection, and you can look up for more information on split beads on this QR code at the bottom of the slides. But basically what we will do in the next step is select fragment sizes of DNA that are between 300 and 700 base pairs long. So these are the ones that bind to the flow cell in the Illumina NGS sequencer. SPRIBITS, it stands for solid phase reversible immobilization. So this approach relies on a concentration of beads and crowding agents. These crowding agents are polyethylene glycol and sodium chloride. This causes that negatively charged DNA binds to the carboxyl group of the bead surface. And the binding depends on the concentration of polyethylene glycol and sodium chloride in the reaction, and the volumetric ratio of the beads to DNA is critical. So as you can see in this gel here, different ratios will bind to different sizes of DNA. So they bind to the larger sizes depending on the ratio of beads and the reagents. In the reaction of these kits, we will first add the beads to the DNA ratio. The first part, we will get all the fragments that are over 700 base pairs attached to the beads. So we remember that we want to get rid of all this. So this will be attached to the beads once we finish the first part of this cleaning step. And we will only keep the supernatant. So we will discard everything that's attached to the beads, so all the fragments that are larger than 700 base pairs. In a second step, we will add beads, and those beads will attach to DNA fragments that are larger than 300 base pairs. So in this case, we want to get rid of the supernatant. We'll have only fragments below 300 base pairs, and we will keep everything that's attached to the beads, which will be everything between 300 and 700 base pairs. We will finally elute the DNA attached to the beads, and we should have this fragment size, which is what we want our library concentration. So we're preparing the ethanol.
Yep. So this is a 80% ethanol, and it need to be prepared freshly on the day, because so now the DNA is going to be bind on the beads. So I think that when we're doing the SPI bead, right? So the DNA, which is actually in the solution, will bind to the surface of the SPI beads, and then the DNA on that beads can easily be eluted into water. So if your ethanol is actually made like last week and then it doesn't have the same amount of ethanol anymore, then you lose your DNA in the washing process. So that's why you have to make it on a day. So once the PCI is done, you can now take the tube out. Spin. So now the library is in the solution. So the template is on the beads, but the amplified library is in the solution. So what we need to do now is to retrieve those library and clean it up. So we need to use the SPI bead to clean it up, and the SPI bead process also remove the primer dimer, which is basically the primers that bind together and then amplify during the PCR, and also removing any artifacts that actually happen during the PCR. So these artifacts usually are the DNA that actually being amplified and misamplified, and therefore it becomes like a larger product. And you don't want to sequence that. The kit, the Nextera Flex kit, already had the SPI beads coming, and it's called a sample purification bead. So I already resuspended the beads. Then we need to add 40 microliters of water. Into a new 1.45 microliters of the SPI beads. Into the tube. Again, use the pipette to fully mix the beads. Now with our library amplification reaction, the beads has now separated from the solution. So we now take 45 microliters of the solution. So this is where the DNA is, and then put it in the SPI bead mix. Mix well and incubate at the room temperature for 5 minutes. On the magnetic stand. So this magnetic stand is the same thing as a magnetic plate, but it's for 1.5 mil tube. And then we can chuck this. And then now we wait for about a couple of minutes just to get the beads to slowly go back, move back
Okay.
to the back of the tube, and then we wait until the solution is clear. Then we prepare another tube. So now that the solution is clear, then we transfer about 130 microliters of the supernatant without the beads into the new tube. And then now just add the undiluted 50 microliter of undiluted beads, and then I'll use the pipette to do the suspend. And then we incubate at a room temperature for another 5 minutes. Incubation is done, then we put the tube back onto the magnetic stand, and it just wait for a couple of minutes so that the bead can gather at the back of the tube. And then now that the solution is clear, then I'm just going to remove the supernatant. So you can actually tip the stand so that the liquid come to the front, and then you can just pipette it out from the front part. So don't touch the bead. And then after that, wash the bead with 80% ethanol twice without disturbing the beads. So this is different from the TWB. Like the TWB, you need to fully resuspend the beads, but for this part you don't have to. And then just leave the ethanol in the tube for like about 30 minutes. And doing another wash.
Yes.
So for after the second ethanol wash, just remove everything. Then close the tube and then do the quick spin, just so that any ethanol that actually stay on the bead will actually gather at the bottom of the tube. And then put the tube back. And then wait for about 1 minute, and then after that use the P20 to remove any ethanol that is left in the tube. So you really need to actually get rid of all the ethanol, because you don't want the ethanol to contaminate the library at the very end. And then after that, air dry the beads for like about 1 to 2 minutes. And then once this is done, we add 30 microliters of nuclease-free water. Or for the if you're using the flex kit, you can also use the resuspension buffer. And basically just add and resuspend the beads in the water. And then leave it sit outside the magnetic stand for 5 minutes. Time, then the DNA will be released from the beads and then get into the solution. So now the 5 minutes is done. Then we just put the tube back onto the magnetic stand, and then just separate the magnetic beads from the solution. Yeah, so you can see the solution is clear. Yep.
Yep.
So now the DNA that we want is in the supernatant. Therefore, we just get another tube. Transfer the sample. Yeah. So make sure that you don't get any beads into the tip, and then transfer into the new clean tube. The library is ready for QC, and that is for qubit and the bioanalyzer.
You will analyze these samples on a bioanalyzer. So bioanalyzer will give you many metrics, and one of them is this graph here. So this graph will show you on the x-axis the fragment size, and the y-axis give you a little bit of a metric of the concentration or the frequency. So this is basically like a histogram, and it'll show you here that most of your library consists of fragment sizes that are between 300 and 400 and 1,000. So you will want to, if you want to clean up more, you can do more on the bead size selection step to just keep the ones that are between 700 and 400. And remember, this is a step that's very important, because these are the ones that will bind to the flow cells. So you want your final library to look something like this.
There are two boxes when you're running the MySeq. One is the flow cell, and the other one is the reagent that actually used for the sequencing. So this one is the flow cell. So you can see the flow cells come in like this.
Yep.
And I don't think you can see. Yeah. So and the wash buffer for the sequencing. So these are the second box. So
And
it's
this
the
is-
reagent for the sequencing.
And this is at minus
Minus 30.
30.
And what it looks like is that so this is the flow cell, and it's actually like you can't go wrong because it's actually said that like load the sample here. Yep. So basically, the library that we just prepared, we have to denature the library and make it at the correct concentration before we just load it onto the sequencer. And basically, this cartridge just have all the other reagents that actually used for sequencing.
Already.
Yeah. Actually, so this is the flow cell. So that's the flow cell.
Okay.
So this is the nano flow cell. So the smallest flow cell. I mean, for the MySeq. And so it only have, like you see, like the part that is not covered by the grating. That is the only part that have the sequencing flow cell color coming. Yep. So this is only like 1 million cluster. Is that 1 million cluster? Like 1 million fragment, and then 2 million read per end. So, so this is a MySeq machine, and I just finished one of the run, and that's why it looked like this. But basically, this is a compartment that you can put the washing buffer, and then this is where you put the cartridge.
So the cartridge will be just reach. Okay. Yep.
So I'll do the wash later. So, yeah, that's. So basically, every cycle, right? It will actually flush the A, and then you take a photo. And then I think this is actually a little bit over cluster.
Okay, so each dot here
Each dot is a cluster. Yeah. So and then like you have the whole like A, C, T, G. Depends on the cycle. Yeah. So.